Hi my dear friends, welcome back to my channel Doctor's Care. Without wasting our time, we will discuss very important topic today, biochemistry of fasting state. As we have discussed earlier about that biochemistry of fed state, what we have studied, let us recap quickly. So what is the hero of our biochemistry of fed state? What is the hormone of fed state? That is, we have studied insulin. Insulin is the hero of all fed state. Now in this discussion, who is the hero here? Glucagon and counter regulatory hormones. What these hormones are doing when our biological system is entering into the fasting state. So broadly, this fasting state has been divided into early fasting, fasting, prolonged fasting, that is called starvation, and prolonged starvation. So when blood glucose levels are going down, when glucose is gone, glucagon will come. When glucose is gone, glucagon will come. Glucagon is a counter, it counters the action of insulin. What we know, like previously what we have studied, insulin is an anabolic hormone. Insulin will stimulate the liver to synthesize the glycogen, to stimulate the same liver to synthesize the endogenous tag which we have stored in the adipose tissue. So lipogenesis, glycogenesis, and this kind of anabolic biochemical alterations we have studied in the biochemistry of fed state. Today discussion is when these glucose levels are going down, glucagon will come. What glucagon will do in altering the biochemical reactions in our body. So already we know when we are in fasting state, so slowly as the time passes, glucose levels will go down. So by the glucose levels are going down, alpha cells of pancreas will release the glucagon hormone, and hypothalamus, because I told you hypothalamus has the glucose sensor. When blood glucose levels are going down in fasting state, it will stimulate the autonomic nervous system to release the epinephrine and norepinephrine. So epinephrine is formed from the adrenal medulla, and cortisol will be released from the adrenal cortex, and not only growth hormone will be releasing from the anterior pituitary. These are all hormones. These are group of hormones which will help in our which will help us in fasting state to maintain the blood glucose level. What is the exact functions of these hormones? Yes, to increase the metabolic fuels in our body. What are the metabolic fuels? Glucose, fatty acids, and ketone bodies. So these we have to increase in the fed state, then our body will be supplied with very good energy. My student, we should know, if the insulin levels remains high in the fed state, what will happen? We know that insulin dependent tissue like heart, adipose tissue, and skeletal muscle, these will keep on taking the glucose, whatever the minor glucose also they will take by the influence of insulin. But we should know insulin levels also should go down when glucose levels are going down. This is very important to understand the core concept. So after this, we have to increase the metabolic fuels in the blood. So how this glucagon will play important role in our discussion. Now we will discuss individually exactly what kind of biochemical alterations which are happening in different stages of fasting. Then we will do the with flowchart with the organ mapping, and we will discuss individual metabolic pathway, and what are the different stages of fasting will use different kind of metabolic fuels basing upon the availability in our blood. So we will start with our discussion without late. Let us start with our biochemistry of fasting state. So biochemistry of fasting state, now these are the stages of the fed fast cycle. Already we know this well fed state 2 to 4 hours after food intake. So that we have discussed very in detail in previous module. Now early fasting, I told you fasting is broadly divided into early fasting, fasting, and prolonged starvation, right? So early fasting 4 to 16 hours without food. I am not taking anything like from 4 to 16 hours, because we know in well-fed state insulin has done so many things. We have synthesized glycogen, we have synthesized triacyl glycerol, we have stored in the adipose tissue, so we know. So if you see the fasting 16 to 48 hours, till 2 days without food, that is called fasting. What is starvation? Exact meaning, biochemical definition of starvation is the prolonged fasting, that is on an average 2 to 5 days without food intake is called prolonged fasting. And prolonged starvation on an average again 5 days without any food, so this is called prolonged starvation. So what is the exactly happening in early fasting? So broadly my friends, these 4 to 16 hours without food intake is called early fasting. What is the source of blood glucose? So I had a example, my dinner at 8 o'clock of at 12 a.m. next morning, or at 1 a.m. in the next morning. Exactly what is happening? What is the source of my blood glucose? Already what I have taken during postprandial, post absorptive, all glucose is going down, insulin levels are going down. So the source of blood glucose in early fasting is, remember, this is hepatic glycogenolysis. Hepatic glycogenolysis. I will explain very detailed example. Let us go back once. Glycosenolysis. Normally in fed state, suppose I am specifically explaining about the liver. I told you liver is like a mother. So mother will always cook, and she will show her love with giving lot of food and all, right? So liver is like a kitchen also, I can say in simple words. See, so example, in fed state, this glucose is converting into glucose 6 phosphate. This glucose 6 phosphate will, we know it is converting into pyruvic acid to, like, by the process of glycolysis. I am not going to detail, we will explain later. So this pyruvic acid will form the acetyl coenzyme. These are the biochemical molecule with the carbon skeletons, right? So this acetyl coenzyme will runs the TCA cycle, will give lot of ATP, that we know this is the normal funda. So now in fed state, excess glucose in the liver, I told you, like, it is storing the glycogen, right? So for this storage of glycogen, this glucose 6 phosphate has to convert into first glucose 1 phosphate, and this glucose 1 phosphate will synthesize the glycogen. Suppose this is the glycogen. Always remember, who is telling to liver to synthesize glucose in fed state? Already we know the hormone insulin. This process is called glycogenesis. But it is not the time to synthesize glucose. I have to break down this glycogen, because our blood, I told you what is the source of blood glucose. This is called hepatic glycogenolysis. What I have stored in the fed state, now I am going to break down this thing. So this same glycogen will come. So by the under the influence of very important hormone, that is called glucagon, and what is called glycosenolysis, breakdown of glycogen to maintain the blood glucose level in the early fasting. So again, by this process, glycogen has to break down into glucose 1 phosphate, and glucose 1 phosphate is converting into glucose 6 phosphate, and this glucose 6 phosphate will converting into same glucose, this by the enzyme glucose 6 phosphatase. So this glucose will go to the blood. Actually this is exactly happening in the liver. Again, no confusion here. See, in early fasting, we have to break down the glycogen what we have synthesized the glycogen in our fed state. Now it is the time to maintain the blood glucose level. When the glucose are going down, glucagon will come. Glucagon will tell to the liver, you please break down the glycogen what you have stored previously. So glucagon will, so here you have to tell that the same glucagon will inhibit the glycogenesis. Same glucagon. So already we know for glycogenesis insulin has to be there, but here insulin levels are decreasing in the early fasting, because when glucose are going down, insulin also go down. That is what we have studied. So this glucagon inhibit the glycogenesis, and this glucagon will stimulate very important process that we know, that is called, what is this? Glycogenolysis. Glycogenolysis. Exactly, this is exact mechanism. Even if you don't understand, no problem, we will study in the metabolism part. So this is the summarizing story of this. So same these access suppose so the stored glycogen is breaking down into glucose 1 phosphate, then glucose 1 phosphate to glucose 6 phosphate, glucose phosphate, glucose 6 phosphate to the glucose, and ultimately the glucose will be released into the blood by the liver through the glucose 2 transporter. This is exactly what is happening. This is the source of blood glucose in early fasting, is hepatic glycogenolysis. Okay, so now I have told you in fed state even muscle has the capability to synthesize the glycogen, but always remember, keep in mind, muscle is very selfish organ. I can say it won't provide the blood glucose in early fasting. Of course indirectly it will help in that regulating blood glucose level in later stages of fasting, but in early fasting only liver is like a mother. Liver is selfless. Liver is providing glucose into the blood by this mechanism. So what is this? Glucagon will come from the alpha cells of pancreas, will tell to the liver to stop glycogenesis, to stimulate. I am stimulating you, you break down the glycogen, that is called glycogenolysis. So why muscle can't provide blood glucose in this test? Can you tell me? Because muscle lacks glucose 6 phosphatase enzyme. I will tell what is this importance. That's why it can't provide the blood glucose. I have told you, for breakdown of glycogen to convert into glucose, you see here, this enzyme is playing very important role in the conversion of glucose 6 phosphate to glucose molecule. But the thing is that this enzyme is absent in the muscle. So what will happen? Glycogen breakdown will happen in the muscle also, but it will come till here only. It can't convert this glucose 6 phosphate to glucose, because due to the deficiency, like due to the absence of this enzyme in the muscle. So this, what will happen in the muscle? This glucose 6 phosphate will go for its own energy. Again this glucose phosphate will converting into pyruvate by the process of glycolytic machinery, and it forms the acetyl coenzyme, runs the Krebs cycle. It will use for its own energy. It is very selfish, but liver is not like that. Yes, liver is like a mother. Again I am repeatedly telling, liver is like a mother, liver is like a kitchen. It will give to others only, right? Easy story. Now, so this is what, so one more point we have to understand, my dear friends. In 16 to 18 hours, whatever glycogen stored in our liver, I told you previously, my liver contained the 2 billion hepatocytes, right? So because it has some limit to store the, because it can't store the glycogen beyond the level. It has some capability. What is the weight of our liver? 1.5 kilograms. This 1.5 kilograms liver can synthesize and can store the only some amount of glycogen. See, this is the capacity. What will happen in this 16 to 18 hours? Glycogen stores are depleted. Glycogen stores are depleted. Total glycogen stores are depleted. Now liver is thinking that what should I do for my body organs, because glycogen stores are depleted. Now how can I maintain the blood glucose level? So our body will choose the alternative biochemical alterations. Now we will go to very important, that is the now next already I have told you, next we are going to the fasting. What are the biochemical things which are happening in the fasting stage and metabolic changes? See, we'll see. I told you 16 to 48 hours without food intake. Without food intake is called fasting state, right? So all glycogen stores are depleted in my liver. Understand? So in the influence of glucagon only, liver has breakdown its stored glycogen and release it into the blood. Muscle can't provide the blood glucose in the early fasting. But if you see, what is the, again same question, what is the source of blood glucose in fasting? Source of blood glucose, always remember, again it is very important, this is called gluconeogenesis. Gluconeogenesis. Genesis means synthesis, gluco means glucose, neo means new glucose synthesis. Who has the capability to gluconeogenesis to do? Again our mother, again our kitchen in our body, that is called liver. Who is the now what is exactly happening to all glycogen storage completed? Now liver is searching alternative pathway, that is called gluconeogenesis, again in the influence of glucagon hormone and counter regulatory. We should not forget the remaining hormones as we have discussed earlier. So what is gluconeogenesis? Synthesis of glucose from non-carbohydrate sources, because carbohydrate sources has already finished in early fasting only. Now we have to synthesize the glucose from new carbon skeleton, because our body is made up of all biochemical. My body has designated, our body has designated in such manner, it has the ability to biochemical alterations of carbon skeleton to give the energetic molecules, that is done by our liver. Example, see, this is the liver. Example, I am taking one hepatocyte. So liver has the capability to synthesize the glucose from non-carbohydrate sources. What are the non-carbohydrate sources? Example 1. Example 1, yes, amino acids. Amino acid, because these amino acids, and second one is the glycerol, and third one is the lactate. We know already lactase always will be produced by our RBC all the time, because it is undergoing the anaerobic glycolysis. Our blood is always readily, like lot of lactate will be available in the blood, and glycerol and amino acids also in the blood. So liver has the capability to take these molecules into the hepatocyte. It has the ability to convert in these non-carbohydrate sources into glucose molecule. So this is called process called gluconeogenesis. Gluconeogenesis, I will explain the same gluconeogenesis in the when we come to the metabolic pathway of gluconeogenesis. But for a while, you trust me, in fasting state our body is searching other metabolic fuel, that is called because it is synthesizing the glucose molecule, whatever the non-carbohydrate sources, whatever the carbon skeletons, right, these are taking from the blood and synthesizing the glucose molecule, again releasing into the blood. This is what it is called, that gluconeogenesis. So always remember, for doing this gluconeogenesis, who is our hero here? Again gluconeogenesis, that is called glucagon hormone. Glucagon level will be increased because the blood glucose level still, like keep on decreasing. We are not taking food for this period 16 to 48 hours. I am not taking any food. So again this glucagon will tell to the liver, actually this glucagon will stimulate the pathways, the enzymes which are requiring for this gluconeogenesis will be activated by the influence of the glucagon hormone. This is called gluconeogenesis. Always remember, for running this mission, this biochemical mission, it is for this gluconeogenetic pathway, we need ATP. Where we are getting ATP? In the liver. Liver do this gluconeogenesis because some fatty acids will undergo beta oxidation, that whatever ATP, because keep in mind, fatty acids are not a source of metabolic fuel in fasting, because the liver has the capability to synthesize the glucose molecule and deliver into the blood. Stage of prolonged fasting, again let us revise what we have studied in early fasting: hepatic glycosenolysis, and in fasting state 16 to 48 hours time we are doing the gluconeogenesis by our kitchen, that is called liver. So in prolonged fasting, exactly what is happening, non-carbohydrate sources also finished in my blood, in my body. So again it is searching some other fuel. My friends, we have discussed earlier in fed state that I told you, we have stored the excess glucose in the form of fat, triacyl glycerol. Before going to know, we should know some basics of this triacyl glycerol. What is exactly tag, students? This is tag triacyl. Tri means 3, acyl 3 fatty acid with 1 glycerol molecule. Suppose this is 1 glycerol molecule, it is attached to 3 fatty acids. Actually this is called exactly triacyl glycerol. Okay, this is the actual tag. So like this we have synthesized the tag in the adipose tissue under the influence of the hormone insulin in fed state. Now where we have stored, I have stored this tag in the adipocyte. Millions and millions of adipocytes are distributed in my body. Now glucagon is the increasing, right? Now example, this is one adipocyte, example one adipocyte. So my adipocyte is full of tag, this triacyl glycerol, understand, it has to break down. Who is the hormone again? This is called lipolysis. We are doing under the influence of same glucagon. I am dividing into 3 fatty acids plus 1 glycerol molecule, 3 fatty acids plus 1 glycerol molecule. Already we know these fatty acids will undergo the process of beta oxidation and will give beta oxidation. I will tell, don't worry, fatty acids are entering into a biochemical pathway that is called beta oxidation. By using this beta oxidation, some process which is called beta oxidation, we don't know anything for a while, we will study later. So by the process of beta oxidation, it is giving abundant acetyl coenzyme, because fatty acids are many carbon skeleton, some 16 carbon, 18 carbon, 20 carbon, these are fatty acids. So we will study in the chemistry of lipids. So this acetyl coenzyme will runs the Krebs cycle and will give the ATP, that we know ATP will give. So but what will happen? So who is for tag lysis, we need an important enzyme that is called hormone sensitive lipase. It is present in all adipocytes, understand. In fasting state, this is in name itself it is saying that hormone sensitive lipase. It is sensitive to some hormone, it will work, some hormone has to stimulate it, then only it will work. Who is telling this hormone sensitive lipase? You break the triacyl glycerol which we have stored previously in fed state. Again our hero, glucagon and counter regulatory hormones also, but mainly glucagon. So this glucagon is telling to the adipocyte, you break the triacyl glycerols. Now our body is in the prolonged fasting. So in early fasting, hepatic glycogenolysis, in fasting state gluconeogenesis, but in this prolonged fasting, students, decreased gluconeogenesis. This is also very important. So no other non-carbohydrate sources also finished. Now the time came to fatty acids. So this glucagon is telling to this hormone sensitive lipase, you please break down the whatever triacyl glycerol you have stored in our adipocytes. So first it will use it's for its own energy. Of course it has to, so because even adipocyte has to survive na, millions of adipocytes in my body has to survive. So after synthesizing, now it is abundant of ATP, now it is utilizing their own energy purpose. But a lot of fatty acids are increasing. These fatty acids will go into the blood. This is the blood. Fatty acids are going into the blood. Now blood is full of fatty acid. These fatty acids will be taken by the different peripheral tissues, all tissues which will use, because remember, for the oxidation of fatty acids requires mitochondria, and that enzyme should be active to do so. Already we know, and it will be taken by the all tissues, like what tissues will need, but brain can't take fatty acid. Always remember, I will tell you later. So these fatty acids undergo the beta oxidation in remaining tissue, those tissue who are taking the which are taking the fatty acid. Again beta oxidation will give lot of acetyl coenzyme A. This lot of acetyl coenzyme A runs TCA cycle, so it will survive by this beta oxidation. That ultimate point is that beta oxidation gives lot of acetyl coenzyme A. Acetyl coenzyme A runs the Krebs cycle, right? This is very important. Only source in the prolonged fasting is that tags get converted into the fatty acid plus glycerol molecule. This is what I want to say in the prolonged fasting. Now our body is entering prolonged starvation. We have studied. So this is the prolonged fasting. This is also called starvation, right? Now this is the prolonged starvation. I told you on an average, I am telling, on an average 5 days without food. 5 days without food. So I can say no glucose molecule, no carbohydrate sources, no gluconeogenesis. Again my all fatty acids also decreased. And sorry students, one more thing you have to know that, see, actually in liver you have to know, in again we are coming 1 minute to prolonged fasting. In this liver what will happen, even liver also are taking the lot of fatty acids which are coming from the adipose tissue. This liver taking fatty acids, these fatty acids will be undergoing the beta oxidation, giving the acetyl coenzyme A. So these acetyl coenzyme runs Krebs cycle, and when the oxaloacetic acid is depleted because it is intermediate of TCA cycle, so these acetyl coenzyme A synthesizing the ketone bodies. Ketone bodies are synthesized in which stage? In prolonged fasting. Keep in mind, ketone bodies are synthesizing. So you keep in mind, even if you don't understand, don't worry. So liver is synthesizing the ketone bodies by using the excess acetyl coenzyme converting into the ketone bodies in the, yes, prolonged fasting. So let us come back into the our topic, prolonged starvation. 5 days without food is called that prolonged starvation. Exactly what is happening, student? So all metabolic fuels in my body decreased, so insulin level is going very low. No insulin molecules also, like in simple words, for sake of understanding, glucagon levels are more and more now. So what will happen, this prolonged stages, so here ketone body synthesis also decreased. Ketone body synthesis also decreased. Very important. So I will, don't worry, like I can say you, maybe you may not understand about these ketone bodies. Don't worry, ketone bodies we will do very great detailed manner. And for a while you trust me, liver is synthesizing the ketone bodies in the prolonged fasting by taking the fatty acids and converting into the acetyl coenzyme. Acetyl coenzyme is giving the ketone bodies. You let it be, leave it this one. You come to this. So ketone body synthesis also decreasing in the prolonged starvation. So exactly what is the source here? I can say that, so muscle proteolysis. Muscle proteolysis. So nothing is available in my blood, nothing is available, so it is switching into the proteins are breaking down into the amino acid. Proteins are nothing but the amino acids, right? So it is leading to, in that stages we can observe that lean nature, or I can say cachexia, lean body, weight loss, very fatigue, these kind of things. So muscle proteolysis leading to the, so these kind of biochemical alterations which are occurring in our body when we are taking when we are not taking food for long days till 5 days or like that. These are the different kind of stages. Now we are going to like in detail about that overview of organ mapping, exactly what is happening. Again I am telling you, liver I am taking, our mother, and so many like foods they are it is making. So liver example, this is example, you believe this is liver. Exactly what is happening in the early fasting, this glucose is converting into, so see, in early fasting whatever the stored glycogen, it is converting into glucose. Understand, glucose. So this glucose is going to the blood by glucose transporter 2, right? So this is called glycosenolysis. So one more fact you have to understand, this is called glycosenolysis. So now glycogen, all glycogen stores are depleted. Now it is taking the from blood only. So from blood it is taking the non-carbohydrate sources like amino acids, lactate and glycerol. I told you glycerol, see here lot of glycerol molecules are increasing. So this glycerol is a carbon skeleton I can use for gluconeogenesis. Again it is telling, it is converting, our liver is converting first it is into the pyruvic acid, pyruvate. So again pyruvate is converting into the glucose molecule, glucose 6 phosphate, glucose 6 phosphate. So this glucose 6 phosphate is converting into glucose by glucose 6 phosphatase, which is majorly occurring in the liver only. So again this glucose is going into the blood. So this is glycogenolysis and gluconeogenesis. What is the third step we have told, understand? And from adipose tissue, this is adipose tissue, lot of tag is breakdown. So lot of tag and giving the free fatty acids plus glycerol. This glycerol I can use for gluconeogenesis. These fatty acids will be taken by the liver. Of course fatty acids will be taken by the other organs which has the capability to run the beta oxidation, right? These fatty acids are giving lot of acetyl coenzyme A. So acetyl coenzyme A can run the Krebs cycle and access acetyl coenzyme by breaking the beta oxidation. These key acetyl coenzyme, by using this acetyl coenzyme is a 2 carbon molecule, 2 carbon biochemical molecule. So if I pack 2 acetyl coenzymes together, this is called ketone body simply. If I am packing 2 acetyl coenzymes here together, or condensation of 2 acetyl coenzyme molecules together, it is called a ketone body. There are 3 kind of ketone body: beta hydroxybutyric acid, acetoacetate and acetone. So I will be telling later when we come to the ketogenesis and ketolysis. Now for a while you tell me, thus these acetyl coenzyme are using for the ketone bodies. Why ketone bodies liver is synthesizing in the prolonged fasting? Because major organs like brain, it never take any metabolic fuel other than glucose. But you know, sometimes if nothing, like you know, something is better than nothing, glucose is not there at all, so it has to take something to survive. So it is a lovely gift from liver is giving to the exclusively for brain, because brain can use the ketone bodies, because ketone bodies can easily cross the blood brain barrier. So these again ketone bodies are going into the blood and it will use it by the brain. Understand? Easy story. If you have understand, well and good, right? Ketone bodies, this is one pathway, ketone body synthesis and ketone bodies. So you have understand this organ, like liver, what exactly what kind of biochemical reactions which are occurring in the liver during the fasting state, right? So, right, now we are going to exactly what is happening in the adipose tissue. adipose tissue. Adipose tissue. This is, yes, adipose tissue. I am taking again one adipose tissue, right? This is one adipose tissue. Right. Exactly what is happening. Again, this is like a revision for us. Lipolysis, we are going to do this here. OI activated, and it is break down the triacyl glycerol into fatty acid plus glycerol molecule. Fatty acid plus glycerol molecule, and these fatty acids run the, we know, beta oxidation. Same thing again. Beta oxidation giving lot of acetyl coenzyme A. This acetyl coenzyme runs TCA cycle, gives energy to own adipose tissue. So lot of fatty acids are increasing. These fatty acids we need for other tissues also. So these fatty acids are coming out of the adipose tissue, and which will be used by the other organs, like example heart, skeletal muscle, and our all body organs except brain, because in brain can't use the free fatty acid. You know why? Because free fatty acid is always bound to albumin when it is in the blood, because this is the lipid, right? Lipid can't travel in the blood, so it has to attach with some carrier protein that is called albumin. You know, one albumin can bind with the 8 fatty acids. A lot of fatty acids are coming, see. So these fatty acids are bound to the albumin, which will be taken by the different tissues, but fatty, because albumin can't cross the blood brain barrier, so brain can't take the free fatty acid. This is what I want to say. So we know that you know, this is some adipose tissue contain the GLUT4. This is insulin dependent glucose transporter. So what will happen in fat state? More GLUT4s will be like expressed on the this adipose tissue. Now it is the fasting state, so no glucose, like very little, like very few glucose transporter spore will be there in the adipose tissue, because glucagon can't translocate through the GLUT4, right? So there will be a less amount of GLUT4 on the surface of the membrane, not allowing glucose to enter adipose tissue, right? Easy. Now hormone sensitive lipase active under the influence of the glucagon hormone, right? Right. So we know exactly what is happening in the adipose tissue also, we know. Now we are going to about exactly what is happening in the resting muscle, resting skeletal muscle. Remember, you know, this muscle example, this is a muscle diagram. I am not good at diagrams. Okay, so this muscle in the fasting, right? So it can use this own glycogen, because it has the capability to synthesize glycogen, and it can use this stored glycogen and as their own energy. This is first glycogen it can use, glycogenolysis, genolysis in the muscle. Second one, because you know liver is supplying the glucose molecule, even muscle also can take from the blood. So this is, and third one, if you see that so you know in starvation it will take the, proteolysis. Proteolysis means a lot of amino acids will come. These amino acids, see, I told you before, muscle will provide the indirect blood glucose. These amino acids will go to liver, and the liver has the capability to convert into these amino acids into glucose in some stages, right? So proteolysis, amino acids, even resting skeletal muscle also will get. So again it has the capability to take the fatty acids also. Fatty acids we know, because muscle has the capability to run the beta oxidation. So this, by beta oxidation, lot of acetyl coenzyme will be forming. By acetyl coenzyme it is giving lot of, it runs the Krebs cycle and getting more ATP. This is exactly happening in the resting skeletal muscle in the different stages of fasting. It can use free fatty acid, it can use the ketone bodies also. Liver synthesized ketone bodies, minor amount of ketone bodies also will taken by the liver. So sorry, resting skeletal muscle, you know, muscle can use everything, because it is selfish organ. It won't provide anything to blood, but it will take everything from blood. But we should you know appreciate muscle in one, it is providing the indirect blood glucose by giving the amino acid converting into pyruvate, you know, it is transported as alanine. Okay, alanine. So this alanine will be taken by liver, it can be used for the gluconeogenesis, right? This is exactly happening in the resting skeletal muscle. So our vital organ in our body, that is called brain. Exactly what is happening in the brain? I told you brain always dependent upon the glucose, always, even if it is fed and fasting. But when the glucose levels are going very down, because no glucose level at the end of day, like at last, what will happen then? Brain what will do? It has to dependent on something to survive, right? So in latter stages of fasting, mainly in the prolonged fasting, that is called starvation, right? So what brain will use? This using the ketone bodies. Ketone bodies only will give 20% of energy, because ketone bodies can easily cross the blood brain barrier. So prolonged starvation, brain mainly depends upon the ketone bodies. What will happen? Brain will take these ketone bodies. I told you what are ketone bodies. This is the condensation of two acetyl coenzyme molecule. Example, this is one ketone body. Example, right? So this is 2 acetyl, this is the 2 carbon skeleton. So example, this is, if I am packing together, these are called, this is called ketone body. Exactly what is happening? This ketone body, by using the ketolytic machinery which is present in the brain, you know, brain has the different engine. Brains are nothing but composed of lot of billions of neurons, right? These neurons has the different ketolytic enzymes. By using this ketolytic enzyme, it is converting this ketone body into, again we know, acetyl coenzyme A. Acetyl coenzyme will runs the Krebs cycle. We know Krebs cycle will gives the lot of energy by the oxidative phosphorylation. We'll study in detail. This is exactly happening in the brain in the different stages of the fasting. Easy story. Of course, when it is in the fed state, we know glucose coming, glucose by running glycolysis, pyruvic acid, acetyl coenzyme A, running Krebs cycle, giving more energy, switching of the oxidative phosphorylation, that is called electron transport chain. These are all very important. Even brain can use the ketone bodies as like 20% of its energy in the latter stages of the fasting. No problem at all, because we will be going to discuss individual, what is glycolysis, individual pathways, how it is regulating with the different hormone, glycogenesis, glycogenolysis, ketolysis, gluconeogenesis, all metabolism we are going to study in detail with very basics. Okay, so but you should know, this is the entire overview of exactly what is happening in the biochemistry of fat state. That's it. Now we are going to sum up these things. And metabolic fuels of fed fast cycle, these are the major organs we have to mug up for our exams mainly, right? Always remember, in fed state, liver uses the more glucose. Glucose is abundant, sir, more than free fatty acid. Glucose is the major metabolic fuel in the fed state. But except I have told you, do you remember, see, hot always prefer free fatty acid. Why? Because our heart is working 24/7, 1 lakh beats per day, because it has more energy. I told you, remember, glucose, complete oxidation of glucose only give 32 ATP. But if I am taking one fatty acid, it is giving the more than 100 ATP. I told you, because one fatty acid is containing the lot of carbons, right? Carbon skeleton, carbon molecule. So it is giving lot of acetyl coenzyme. One glucose molecule can give only 2 acetyl coenzyme A. But remember, one fatty acid can give lot of acetyl coenzyme A. Acetyl coenzyme A runs the Krebs cycle, that we know. So heart is always dependent upon the free fatty acids, even if it is having the glucose as major metabolic fuel. Even of course it can use glycolysis, also will be happening simultaneously with the beta oxidation, right? So you no need to confuse. But 24/7 it is working all the time. It needs more and more ATP. So it needs more acetyl coenzyme. More acetyl coenzyme is coming from the beta oxidation of the free fatty acids in the heart, right? Easy. So now what brain will use? Glucose. It always prefers glucose. And muscle, what it will prefer? Glucose in fat state. RBC always glucose, and adipocytes glucose and free fatty acids. First it will prefer the glucose. Adipocytes taking the glucose in the fat state, right? So in early fasting and fasting, if you see that, like normally in imagination, normally liver will take the free fatty acid. Why can't it use glucose molecule? Because it can't take. It is, even if it will take the free fatty acid, it is like a lovely nature, right? It will synthesize the glucose, whatever glucose it will give to the blood to other organs to survive. It will take free fatty acid and lot of oxidation, acetyl coenzyme runs and uses for its own metabolic energy, and as well as giving glucose molecule to other. Heart always use free fatty acid, then glucose. Brain glucose. Muscle free fatty acid, because it is the fasting state free fatty acid coming from the adipose tissue. And RBC will always use glucose. My question is that why RBC can't use free fatty acids? Always remember, my dear friends, RBC doesn't have mitochondria. If I want to break down the fatty acid, I told you some process which is called that beta oxidation mission, that cutting enzymes. Understand, for cutting the fatty acids I need some missions, right? That missions are present in only in mitochondria. Beta oxidation are always present, like occurring in the mitochondria, but my RBCs, the poor stuff, it doesn't have our mitochondria. So free fatty acids can't use it by the RBC. It can use only, even if you are giving lot of things, even anything other than glucose it can't take, because it doesn't have mitochondria for occurring some, but even see why glucose, even this glucose can't undergo complete oxidation also, because complete oxidation also requires mitochondria. That is called Krebs cycle. Krebs cycle is a part of mitochondria. That missionary enzymes of Krebs cycle is also present in the mitochondria. So that we know, right? So adipose tissue in that fasting state also free fatty acids it will prefer than glucose. But in starvation or in prolonged starvation, mainly we'll see the prolonged fasting, that is called starvation. So liver can use the amino acids. In starvation, I told you muscle is providing the amino acids to the liver. I told you, because in that way it is providing energy, like indirectly, not directly. So again it will be used as glucose after coming from liver, Cori's cycle and Cahill cycle, right? We will be discussing later. So always remember, liver is synthesizing the ketone bodies, but it never use ketone bodies. Why? We'll discuss later. Okay, I can say, for our sake of understanding, it is lovely nature. Understand? That's why it is not using. It is giving a lovely gift pack to the brain to use these ketone bodies in the starvation stages, right? And heart always prepare free fatty acids, and it can use, because glucose is absent in starvation, so ketone bodies it can prefer for survival, right? So brain always prefers glucose. If glucose, even minor one molecule of glucose is present in the blood, it can take, right? And ketone body, we know 20% of energy is deriving from the ketone body. In muscle free fatty acids, we know ketone bodies also it can use. RBC always poor stuff. But my question is that you can ask me, so you said no glucose in the latter stages of fasting, that is called starvation. So yes, if that glucose is not absent, RBCs undergo hemolysis. It will die. It will survive in the presence of glucose. It will tolerate for some time. If you give what kind of metabolic fuel with lot of things, ice creams, give whatever you give, it can't take. It needs only glucose, but glucose is absent. RBCs undergo the hemolysis. That's why in starvation we can observe the lean, like lot of pallor. You can observe that human, those who are not taking food more than 5 days, you can easily observe what kind of things which are happening in their body. So hemolysis will happen, because glucose is not there at all. If it is there, it will take, otherwise that RBCs undergo hemolysis. Normally in adipocytes, free fatty acids and ketone bodies which can be synthesized by liver also use it. So 2 points I want to share here. And RBC never use ketone bodies. This point you have to keep in mind. This is one thing. And one more thing, completed our biochemistry of fasting state, but we know the what is the exact functions of the glucagon. So keep in mind, I'm telling very fast, I know, but very less time also, right? So first it is inhibiting the glycogenesis. It's stimulating the glycogenolysis. And right, activation of gluconeogenesis, and we can say increasing or stimulating the lipolysis. Of course it will inhibit the lipogenesis, right? And lastly it will stimulate the ketogenesis, that we know, right? And yes, we know it is stimulating the beta oxidation also, by fatty acids. So these are all kind of biochemical reaction which is occurring in the influence of, because I can say glucagon is purely catabolic hormone. Catabolic means something breakdown. We are breaking down the glycogen. We are I can say again, you may ask, we are synthesizing the glucose. This is one exception where the acting as anabolic phenomena, I will tell later. So these are all kind of you understand these things, why glycogenesis inhibiting, glycogenolysis is stimulating in the liver by the under the influence of glucagon. And we will study the same thing whatever we have studied in first video, second video. Now this is third video. We are going to discuss in detail of each and every metabolic pathway. This is what I want to say. If any mistakes are there, you please forgive me, my dear friends. I am improving myself from you only, and thank you so much. And we will from next videos we will be doing about the chemistry of carbohydrates and metabolisms of carbohydrates in detailed with simple basics.